second form of cell division. Of course, it occurs in non-somatic cells, otherwise germ cells, which are the cells involved in sexual reproduction. Now, before I continue, I need to chip in something that is very, very essential for your understanding of the topic. In every cell, for every species, there is a fixed number of chromosomes in the nucleus of the cell. That number is called the diploid number. And it is represented by 2n. Now, it is diploid because for every chromosome set, there is a double for it. One gotten from either parent. So you have each of these chromosomes from your mom and from your dad. So for every set, you have double. That's why it's called the diploid number. Okay? So for a cell that is not divided, that is a cell in interphase, that's what you have, 2n. And don't forget, if you will recall, I said the interphase is divided into 3 parts. You have a G1, you have an S, and you have a G2. So for a cell in interphase, a cell not divided, the number is 2n. But don't forget, I also said something about the S phase. I said the S phase is a stage where the cell would duplicate the genetic material, would synthesize and make it double in anticipation for a division. So at S phase, this number will be doubled. And what the cell now has is 4n. So going into G2 and into division, the cell has 4n chromosomes. Always bear that in mind. So I'm taking this again. I'm saying for every species, amount of chromosomes you have in the nucleus for a non-dividing cell is the diploid number. But don't forget, at S phase, it will duplicate and it becomes 4n. So a cell that is dividing has 4n chromosomes. Is that okay? Now, in mitosis, what I just finished, we divide this into 2 equal parts, one for each daughter cell. So it restores back the diploid number. So this is a parent cell that has 4n. It then divides into 2 cells, each with 2n. This is what happens in mitosis. This is what happens in mitosis. So the 2 daughter cells formed in mitosis are diploid. Okay? Now let's look at meiosis. Let's look at meiosis. Let's look at meiosis. I've already said that in meiosis, this occurs in germ cells. In germ cells. And you form 4 daughter cells. 4 daughter cells. And I also said something about genetic diversity, that there is genetic diversity. Last but not the least, there are 2 divisions in meiosis. 2 divisions. 2 divisions. Is that okay? These divisions, I will name them Meiosis I and Meiosis II. Now let's look at the detail of what happens in these divisions. Okay? Do not forget, a cell that wants to divide has how many chromosomes? 4n chromosomes. Okay? Now let's look at what happens. The stages are still the same. So for Meiosis I, you have P mat again. For each of them is 1. Is that okay? So you have prophase 1, you have metaphase 1, you have anaphase 1, and you have telophase 1. In division 2, you still have the same divisions, P mat, but this time 2. So this is for Meiosis I, and this is for Meiosis II. The same procedure. You have prophase 1, metaphase 1, anaphase 1, and telophase 1. The same divisions you had in mitosis. They repeat themselves here again in meiosis. Just that the division occurs twice. Is that okay? So let's look at it and see. In prophase of the first meiotic division, everything we already said in mitosis occurs. So this is the cell membrane. Let's look at interphase. This is the nuclear membrane with chromatin, thread-like chromatin. So this is interphase, a cell that is not divided yet. Okay, in prophase 1, pro P1, in prophase 1, the cell membrane is intact, but the nuclear membrane will break down, so I don't have a nuclear membrane. The same thing that happened in mitosis. This chromatin will condense into chromosomes, the same thing that happened in mitosis. So what I now have is condensed chromosomes, condensed. condensed chromosomes, condensed like that. Again, centrioles will appear and start migrating. So I have a migrating centriole. I have a centriole migrating. I have a centriole migrating. Okay? Spindle fibers will also start forming. This is prophase 1. Then in metaphase 1, let me call it M1, in metaphase 1, I have the cell. The centrioles have already gotten to the ends. Spindle fibers have been formed. Just the same procedures that happened in mitosis are repeating themselves. But the difference here now is this. The difference between mitosis and meiosis now is this. In mitosis, what you had arranged were chromosomes made up of chromatids connected by centromere. This was what you had in mitosis. The case is different in meiosis. What you have arranged, yes, are chromosomes, true. But at this stage, what will arrange themselves are homologous chromosomes. What did I call them? Homologous chromosomes. What will arrange themselves are homologous chromosomes. Now, you might not understand the concept of homologous chromosomes, so let me tell you what that is. Homologous chromosomes are chromosomes of the same set of genes, just that you got them from either of your parents. So if it's chromosome 1, I'm talking about C1. I got one from my mom, I got one from my dad. So these two are homologous chromosomes. They have the same set of genes from head to toe. The only difference is I got one from my mom and I got one from my dad. So it is this homologous set of chromosomes that will arrange themselves at the equator, at the equator, like that. So this is metaphase 1. And these, do not forget, are homologous chromosomes. Not chromatids. Now, each homologous chromosome appears like this. So I have a homologous chromosome like that. Okay, made up of 2 chromatids, clearly connected at centromere, and then another homologous chromosome. So these are a pair of homologous chromosomes, one from mom, one from dad. The same chromosomes. They have arranged themselves at the equator. Now, if you look at this carefully, these chromosomes meet at this point and at this point. Okay? At this point and at this point. These places are very important for genetic diversity because there is something called cross-linkaging. Cross-linkages. Now, some parts of this chromosome will break up and attach to that of this. Some of these will break up and attach to that of this. And then eventually the chromosomes you have will mix up. This is what adds genetic diversity. This is what adds genetic diversity to meiosis. Because there is a genetic recombination, as you can see in meiosis 1, when homologous chromosomes arrange themselves in the equator, metaphase 1 of meiosis. Is that okay? Then in anaphase 1, the same thing that happens in mitosis occurs. This homologous pair will separate. So I have homologous pairs separating. So to this other side, I have anaphase 1, A1. I have centriole, I have chromosome. They are all separating. Is that okay? They are all separating and moving towards the ends. Now in telophase 1, they arrive. In telophase 1, in T1, now in telophase, the homologous chromosomes have already arrived both ends of the cell, like that, just like that. Clearly, the opposite of what happened in prophase will again happen. A nuclear envelope will form, they will decondense, another. Is that okay? And then the cell will divide. Eventually, this is not complete yet, and then it ends. So this is the very first step of meiosis, meiosis 1. there are 4 stages in PMAT. 1. First prophase, first metaphase, first anaphase, and a first telophase. But do not forget what divided here: homologous chromosomes. Also, do not forget this is the stage where you have genetic recombination, creating genetic diversity. Is that okay? Now, the second phase of meiosis again has 4 phases: prophase, metaphase, anaphase, and telophase. But the difference between this second phase and the first phase is that whereas in the first phase, homologous chromosomes separated, in the second phase, chromatids will separate, just like in mitosis. So we start the process anew from prophase to telophase. So for prophase 2, again, the nuclear membrane will disappear. So for each cell, so I now have 2 cells this time around. So for each cell, the nuclear membrane will disappear. The chromatin will again condense back into chromosomes, this time having chromatids. So each chromosome now has chromatids attached at the centromere like that. Each cell, each cell, each cell, each cell, like that. Centrioles will again form and start migrating. Centrioles will form and start migrating. Spindle fibers will start forming. I've said this like 1,000 times already. So this is basically what happens in prophase. Nuclear membrane disappear, condense back, centrioles form spindle fibers. That's prophase 2. Then metaphase 2, M2. The spindle fibers have been formed. Centrioles are at both ends. Centrioles are at both ends. Centrioles are at both ends, and they are formed spindle fibers again. Spindle fibers again. Okay? Once again, the chromosomes are arranged at the equator. The chromosomes are arranged at the equator. This is always in metaphase. This is always in metaphase. This is always in metaphase. Okay? This is always in metaphase. This is always in metaphase. But don't forget, this time around, it is not homologous chromosomes. It's just individual chromosomes made up of chromatids attached together at their centromere. Okay? So this is basically what happens in metaphase 2. The chromosomes appearing as chromatids will arrange themselves in the equator of the cell, attached to the spindle fibers at their centromere. Then anaphase 2, A2. These chromatids will separate. The chromatids will separate. And then what you have is a big cell with separating chromatids, like that. With separating chromatids. You have big cells with separating chromatids. Okay, so this is anaphase 2. Eventually, you move on to telophase 2. Move on to telophase 2, T2. In T2, the chromatids will reach the ends or the poles of the cells. They reach the poles of the cells where a nuclear membrane will reform. They will decondense back to chromatin, centrioles will disappear, spindle fibers will disappear, and eventually again cytokinesis will occur. Cytokinesis will occur. And what you now have is 4 cells. You now have 4 cells. You now have 4 cells like that. You now have 4 cells. You now have 4 cells. Let me draw this properly. You end up having 4 cells. You end up having 4 cells. You end up having 4 cells. So this is meiosis. So meiosis, the form of reproduction that occurs in germ cells and forms 4 daughter cells, has genetic diversity and has 2 divisions. That is it. So in summary, we looked at cell division, and these are the 2 main types, meiosis and mitosis. I said meiosis occurs in germ cells and forms 4 unidentical daughter cells, thanks to genetic recombination that occurs in meiosis I by cross-linkage. I said there are 2 divisions, and that's meiosis I and meiosis II. Meiosis I and meiosis II. Okay? I'm saying that it's actually very important in sexual reproduction. While mitosis occurs in somatic cells, it's very important in growth and tissue repair, and usually forms 2 identical daughter cells. Another difference is that daughter cells formed in mitosis are diploid. That is, they have 2 N chromosomes. Whereas those formed in meiosis are haploid. They have only N chromosomes. They have only N chromosomes. Is that okay? So this is the summary of cell divisions. Now let's look at a few questions. Okay? Let's look at a few questions. I saw this one in jam past question. I can't remember the year all the same. But then this is what the question says. The question says, a species of plants has 26 chromosomes. The question they're asking is, what will be the number of chromosomes in a cell that is about to start mitosis? The options are A, 26, B, 13, C, 52, and D, all of the above. Now clearly, the answer to this question is going to be 52. And the reason is simple. You see that for normal cells, they are diploid, that's 2N. And they are saying that the normal cell is 26, that's 2N is 26. Okay? You see the cell that is about to start mitosis must have undergone interphase. And we said interphase has G1, S, and G2. I would say that in S phase there is synthesis and a duplication of the genetic material. So for a cell to want to start mitosis, it must have duplicated the genetic material prior to that. So it must have duplicated 26 and eventually now has 52 before it starts mitosis. Okay? Another question. The somatic cells of a goat, have 100 chromosomes. Somatic cells of a goat have 100 chromosomes. How many chromosomes will be in a sperm cell of a goat? That's the question. And a somatic cell of a goat has 100 chromosomes. How many chromosomes will be in a sperm cell of a goat? A, 100. B, 50. C, 200. D, 25. Well, the answer to this question is 50. And the reason behind that is very simple. A normal somatic cell, not dividable, has 100. That means 2N is 100. And we are asked to find the number of chromosomes in a sperm cell. A sperm cell is a germ cell. And germ cells are formed by meiosis. And we said that meiosis halves the number of chromosomes, and what they form is haploid cells. So it means a sperm cell formed by meiosis does not have 2N. It only has N, haploid. And that means N is 100 divided by 2, and that's 50. So yes, the sperm cell of a goat would have 50 chromosomes. 50 chromosomes. Okay? Now another question I saw that is very interesting is this. Question 3 from Jam Pass Questions. The ovules in the flower of a hibiscus have 5 chromosomes. How many chromosomes will be in a mesophyll cell? The mesophyll cell of a plant is a somatic cell. and so would have 2 N chromosomes. But the ovule in the hibiscus flower is a germ cell and has N. So ovules are germ cells, whereas mesophyll cells are somatic cells. So ovules have N, and from the question, that N is 5. And now they are asking us how many are there in the mesophyll cell. Mesophyll is somatic, and that's 2N. So that would be 2 times 5, and that's 10 chromosomes. Okay? So for many more questions like this, you could refer to your JAMB past questions and your different school post-UTME questions. And do not forget to hit the subscription button and also hit the notification icon so that you get notified on any of our subsequent videos. Also, do not forget to share with your friends who will find this very useful. Comment any difficulties you have or any topics you recommend us to handle on the comment section below. And thank you.